All right. Welcome. We'll get started as folks trickle in. Luckily, the session is recorded, and so we might have folks revisiting this a little later. Thanks for being patient. Just for a couple minutes, this is our first webinar for the winter quarter here at UC Davis, and so we're just trying to get things all aligned. So thanks again for being patient. My name is Eric Chu. I'm a faculty member in the Department of Human Ecology in the Community and Regional Development Program here at UC Davis. I have the privileged role of being one of the co-directors of the UC Davis Climate Adaptation Research Center. And we're very happy and grateful that another co-director, Dr. Greg Monroe, has agreed to launch our winter quarter seminar series, speaking a little bit about his work on plant genomics on a changing planet. But before we begin, just a couple sort of ground rules as we sort of convene here, and all attendees right now are in listen-only mode. However, it doesn't mean that you can't ask questions or anything. I ask that you use the chat function to send these questions on to the group. Our wonderful student assistant, Sophia Depew, is here to monitor the chat room. And so please send any questions, comments, anything that needs troubleshooting to Sophia in the chat box during the session. I'm going to ask that Dr. Monroe sort of go through his presentation in its entirety, and then we'll turn to the questions at the very end. And so the chat box will be a way to sort of collect all the questions, and then I'll do my best to manage the questions as we go along. Everybody is muted right now except for me and Gray, and so that's great. Please refrain from unmuting yourself during the presentation itself. But once we get to the question and answer session, of course, you can unmute and turn on your video if you choose to. All right, with that being said, I'd like to introduce Dr. Gray Monroe. Gray is a faculty member here at UC Davis in the Department of Plant Science. He joined us in 2020, and before that, he completed a PhD degree in ecology from Colorado State University. Dr. Monroe is an expert and is really interested in developing a deeper understanding of how plants adapt to their environments and to address practical challenges of climate adaptation in agriculture through genomic discoveries. And I'm sure we'll learn a whole lot more about that in the presentation today. So with that, Dr. Monroe, I'm going to pass it on to you.
Okay. Hello, everybody. Thank you. Thanks for the invitation to speak. Thanks for being here, those of you joining. I'll share my screen. You guys seeing my my slide here?
Yes, we do.
Great. Yeah, thanks for the introduction, Eric, and pleasure to be here. So our lab is interested in understanding how plants adapt to their environments and the genetic basis of that. I've thrown up this dramatic image to remind us of the challenges that we face around the world, but a lot of that is quite acute and obvious to us, those of us in California. An image of a wildfire raging just across from a vineyard, which highlights the challenges of climate change for our agricultural and ecosystems here in California and around the world. So today hopefully I will give a very broad overview of what we do, what we're interested in, and try to convince you that there's value to be gained from studying natural diversity in both wild species and in crop species if we want to develop a deeper understanding of adaptation that has happened on an evolutionary scale, which gives us a better understanding of how plants adapt to the challenges of their environments, but also hopefully provides a research framework that can be applied to making our agricultural systems more robust in the face of climate change. Before I get into it, let me first just acknowledge the folks in my lab. Here's not everyone, but a number of them and people who have worked on the things I'll talk about briefly today. Some collaborators who have played an important role in some of the projects that I'll briefly touch on. various sources of funding that have provided support for the work I'll talk about. And then I'd like to take this as an opportunity to acknowledge the land we're on here in Davis, which has traditionally been home of the Patwin people. So we're grateful to all of these people and all these collaborators, et cetera, for their work that I'll talk about. Unfortunately, we have to start with the bad news, which is that many regions around the world are predicted to experience a reduction of crop yields as a consequence of changing climates, changes in land use, et cetera, resource availability. Here's a map of the world showing the percent changes in yields of many crops in different regions over the next decades and over the next century. And so this is something we should be worried about, which is something we should be thinking about. And because a lot of this reduction is predicted as a consequence of the mismatch between the plants that we're trying to grow and the environments that they'll see in the future, this really comes down to a lot of this phenomenon is a product of pairing organisms to the proper environment, because the crops we plant today are adapted for the climates of today and not necessarily for the climates of tomorrow. So one of the goals of our lab is to try to understand how climate adaptation works so that we can predict and make better decisions about how to make crops for the future. So it's not just crops that are imperiled by climate change. Of course, biodiversity is threatened. So here's a map showing the predictions of regions around the US that are predicted to see losses in biodiversity. And hopefully today I'll convince you that this is something we should care about, because biodiversity provides a source of information that we can use to better understand things like adaptation. If we want to understand and if we want to make crops that are better suited for the environment, having biodiversity that we can tap into as a source of information is something that we should value and protect, because it's valuable information that we're losing if we lose biodiversity. So both of these challenges, both in agricultural systems and in natural ecosystems, reflect or raise the question of how are we going to adapt our cropping systems to future climates? How will organisms and the environment adapt to changes in climate? But I think it's worth taking a moment to remind ourselves that species on this planet have been adapting to the climate for, well, since the origin of life on Earth. So adaptation to environment is a fundamental phenomenon, process, something we can observe in species and populations of organisms. So adaptation from an evolutionary sense is something that we've known about for a very long time. So all the way back to, if we go back to Charles Darwin's book on the origin of species, this is noted quite clearly that species adapted to the climate of their home we see adaptation. So if you take a species from an Arctic region and a temperate region and you try to grow them in a tropical environment, they won't do well, or the other way around. So there's this phenomenon of local adaptation to climate within species, between species, that has been known and discussed for a very long time. And so by studying these processes and by developing a deeper understanding of their genetic basis, we're hoping that we can apply this knowledge to make better decisions in the face of climate change. So in this particular case, Darwin is speaking about differences between species, but we also know that there's differences within the same species. And this has been something that's been documented for a very long time. So one fun example is from Carl Linnaeus, who's famous for developing the nomenclature system of species. And he would travel around Europe, make observations of different species. And even 300 years ago, he is traveling through Europe and notices that there's this very strong gradient that in this particular species, you see that they flower at different times, depending on where they're from a sort of across a climate gradient from north to south. So things in the south, they flower earlier in the species, but they flower much later in the north. And over the centuries that have followed, lots of experiments have been done to confirm that these types of phenomenon are largely genetic in origin. So there's genetic differences between populations from the south and populations from the north, that these differences in things like flowering date are actually attributed to differences in their genetics. So they've been these organisms, this plant has been adapting to this to different climates, and the outcome of that is changes in these phenotypes and what we observe flowering, for example, but also in their genotypes, the genetics. So you can even see such phenomenon happening right here in California. So if you went on a long enough hike through the Sierra Nevadas, if you started very low and went very high, you might observe things like this is a classic system that's been studied in California quite a lot, called Yarrow. And if you were to be at the base of a mountain range, you might find this same, looking at the same species, you would see it maybe flowering down low, but at the top of these mountain ranges, you would find them, still dormant, and there are differences in sizes between these different things. And there's been lots of experiments to test the genetic basis of such observations. In fact, some of the very origins of the first kind of serious experiments on climate adaptation in plants was done here in California. So this famous trio, Clausen, Keck, and Heisey, were based in California at the Carnegie Institute, and performed this this experiment where they took this plant species that they collected all the way down from the coastal region to the very tips of the Sierra Nevada mountains. And what they observed was a very robust, and they could repeat this over and over. There was this observation that when you collected seed from all these different locations and you grew them in the common garden, so you grow them in the same environment, and by doing so, you can really determine the effect of genetic differences. And what they found is that indeed, populations of this species from the most northern environments, or sorry, the most high elevation environments, for example, were much smaller in size, whereas things from low elevation tended to be larger in size, even when grown in the same environment. And by planting in different places and lots of different experiments, they were able to demonstrate that this variation is a reflection of genetic differences between these different groups and is adaptive, meaning that being small at high elevations is actually an important trait. If you try to grow something that tries to be big up here, it won't survive. And so this is an adaptation. So we're really observing an adaptive gradient from the coast all the way to the tops of mountains that has resulted in genetic differences between these different populations. So this is kind of the fundamental paradigm by which we start thinking about studying climate adaptation in plant species. We look at differences between populations collected across different climates. We look at differences in their phenotypes, so the traits that we can see. And we now, with the ability to look at DNA at the sequence level, we're starting to characterize what is the DNA basis of these variations. What is the genetic basis of this evolutionary adaptation? So this is what our lab does. I'll give a couple of brief, we won't get into too much of the weeds, no pun intended, about what we found, but hopefully this gives you an overview of kind of why studying the genetic basis is useful, and give you some sneak peek into some of the projects that we've started since joining at UC Davis. So first, let me just give an example of some work that we've done. So this is a species, it's a classic model system for ecological evolution. It's called Arabidopsis thaliana, and it is distributed largely throughout Europe, but it's distributed across diverse climate gradients. So here's a map of its distribution. Every one of these little crosshairs on this map is the location where someone went out and actually collected seed from a plant. That seed was grown in a lab. It was the DNA was extracted and the whole genome was sequenced. So we know all of the base pairs of the DNA of every one of these individuals. There's about 1,100 different accessions collected across this climate gradient. So I've colored this map according to a measure that we used that we call the drought timing index, which is an indication of what time of year plants tend to experience water deprivation. And we were interested to ask, do we see variation across these climate gradients at the genetic level? Are there examples of genes where we see variation in genes that are predicted by what climate an individual comes from. When we do this, we found a lot of genetic variation that seems to be important. So this is exciting. So this is a sort of a visualization of the genome of this little plant, Arabidopsis. We have each of the 5 chromosomes here drawn, and we've kind of marked with these little dots. These are the location of different genes that we find significant. We find significant variation at these genes across these climate gradients between regions that have spring and summer droughts. So these provide interesting candidates for thinking about for climate adaptation. So we can start asking questions about, do these genes do anything important for phenotypes? Do they change things like traits? And what we've done is we did experiments to check what these genes do. And as just sort of the final punchline here is that what we've discovered is that by looking at, I'll explain this figure. So each one of these crosshairs is in one of these accessions. On the y-axis, we have the flowering date. So we grew these plants, they were in a common garden, and we asked how long does it take them to flower from the time of germination. And what we found is that we could predict the flowering based on the number of alleles, the number of genetic variants that were associated with spring drought environments. So accessions that are here, for example, they flower very late, and they tend to have the genetic variants that are associated with these environments where droughts tend to happen very early during the growing season. And we've done experiments with these genes to show that they do, in fact, change flowering time accordingly. So why does this matter? Why is this useful? What this demonstrates is that we can look at climate gradients. We can study natural populations from across climate gradients, which allows us to discover genes that are important for traits such as flowering time that confer some adaptation to something like drought. So by studying natural populations of plants from diverse environments, we can gain an understanding of things like the relationship between some trait like flowering time and drought, but also start to gain insights about the genetic basis of that, which is useful both from a fundamental perspective, but also starts to inspire questions about how can we apply these kind of approaches to crops, for example. How can we apply this knowledge to develop crops that are better adapted for environments? So in this particular case, this is like I said, this is a model system. It has what we would describe as a small genome. So it was one of the first species for which we had a whole genome sequence data available at this scale, and which is why it was kind of a pioneering species for this kind of experiment. However, in case you weren't aware, we are in the midst of a very much a revolution in genetics and genomics and DNA sequencing, which means DNA sequencing, we're referring to just technologies that allow us to actually figure out the base pair, the base pair composition of an organism's genome. Maybe you've submitted your own DNA to something like 23andMe as a way of learning something about your own DNA. But this and similar approaches have become possible because we're in the midst of this incredible transformation in this field due to the reduced cost of DNA sequencing. So here on this orange line shows the cost of DNA sequencing per million base pairs, and note that the y-axis is on a log 10 scale. So tremendous changes in the cost of DNA sequencing, but it's also worth to put this in perspective. We have this white line. This is called Moore's law. This is the line that describes, for example, the change in computational power. So starting a long time ago, someone noticed that we were doubling the processing speed of our computers every 2 years. And if you can appreciate how powerful your cell phone is compared to something in the 1970s or even 10 years ago, that follows Moore's law. But the point of this is to show that the cost of something like DNA sequencing has far surpassed something even as powerful as our computational power. So it really has transformed the field of genomics in a profound way. Because what it's allowed us to now do is to survey genetic variation in different species at a scale that even 15 years ago would have been hard to fathom. So here's some Here's some just examples of data where this is now being done in crop species. I've just made this little figure where we can see the number of, these are different. On the x-axis, we have date, and these little tick marks represent various publications of different species where lots of genomes have been sequenced. And what I mean by that is there's been, this is, so for example, cassava, this is a crop I'll talk about in a minute. There was a project in 2017 where 241 genomes, so this means 241 different varieties of cassava, their entire genome was sequenced. We have in rice over 3,000, cacao, beans. This date is here, but this exponential curve has only continued to go more vertical in the last few years. So we really are seeing an ability to study genomic variation in a way that's truly transformative. Okay, so I'm going to take a moment and I'm going to tell you briefly about 3 projects that are happening in our group. And I think this is just going to be a snapshot. Most of these are actually rather new projects, so you'll see kind of what we're doing. But mostly it'll just be an overview of where we hope to go in these different species. So we'll talk about wheat. This is a super important crop if you like your bread, if you like your hefeweizen and beers. It's actually the leading source of protein in human food around the world from vegetable sources. We'll talk about cassava. This is a very important crop for especially in a lot of marginalized communities. It's actually an incredibly high yielding crop. It has more calories per acre per season than maize and rice, et cetera. So a very important source of calories for a lot of people around the world. And then we'll talk about pistachio, which is very important for California agriculture. California is the largest producer globally. If you've eaten a pistachio, it's almost certain that it came from California. It has an incredibly low carbon footprint compared to other sources of protein. And depending on how it's grown, can use less water than other nuts. So these are why we study these crops. They're important, and they also provide opportunities for us to ask questions about climate adaptation in these different species. Okay, so wheat, wonderful system, fun to work on. We are taking advantage of something that, if you didn't know about, is very cool. The USDA maintains something called the Germplasm Resources Information Network. So this is a map of, obviously, with little, each of these little marks is the location of a place where there is a genetic, there's a gene bank. It means that there's lots of seed stored somewhere. Someone is storing a repository of genetic diversity. We'll talk about some genetic diversity that's maintained here in Idaho at the National Small Grains Collection. So what I mean by a seed bank is it looks something like this. It's a room, usually it's cold, and there's in these long shelves with little inside of each one of these shelves, there's packets, and inside of each one of these packets, there's seed. And so every one of these packets represents some different genetic variety of some species. As I said, we're interested in wheat. We happen to be studying a species of wheat called Triticum monococcum. It's also called einkorn wheat. And we're specifically interested in collections of wheat that have occurred. These are wild. This is actually a wild wheat. We're studying a wild species of wheat that has not been domesticated because, as I said, as I sort of introduced, our interest in these types of systems is to understand things like climate adaptation. So by studying a wild relative of something that we eat, we can gain insight into its adaptation to climate. Okay, so all these little packets of seed, where did they come from? These seeds didn't just magically appear. They were collected. So here's an example. Someone actually went out to an environment. So here's this is somewhere in Turkey, I believe. And they're just literally looking for this plant. They're going and walking around and looking for specimens of this species, this organism. And when they find it, this has largely been done by many different people. The USDA has put a lot of effort into these kind of collections. Here's an image of someone actually collecting this species, wild wheat. This is from like the 1970s. And so they find this species, they collect the seed, they put it in a packet, and it's secured now and maintained by the USDA in their collection. So we're studying a collection of this wild wheat species that has been collected from across the native range. So this is where this species naturally lives. largely in Turkey, Syria, Iraq, Iran. And this is exciting because these different populations of this species come from actually rather diverse climates. So this is actually a pretty diverse range of different climate environments. Here's the pictures. We see diversity, for example, at the seed color level. But what we're interested in is understanding whether or not this variation across climates has resulted in climate adaptation. So are these different populations adapted to these different environments or not? I'll show you one slide with some results looking at phenotypic diversity, so trait variation. And I'll say that these genomes have been sequenced of all these individuals in collaboration with a number of different groups, and we're now studying the genetic basis of adaptation. So what do the phenotypes look like, though? So we've been able to gain some interesting insights from looking at these populations. For example, you may know this or not, that wheat often comes in two different categories. So we have spring wheat. This type of wheat can be planted in the spring and it will flower. So you can plant it in the spring and it will produce seed in the same season, whereas winter wheat requires that it's planted in the fall and it has to go over the winter in order to flower in the following season. These two different types of wheat are very important agronomically, depending on what kind of environment you want to grow something in. And it turns out that that variation exists also in natural, remember, this is wild, these are wild species, wild populations of wild species. And what we find is that there's significant differences between whether something is a spring wheat variety or a winter wheat variety, depending on the type of seasonality of precipitation. So what this means is that places that have that adopt a spring type growth habit tend to come from places that have a much more variable environment in terms of precipitation. So what we think is going on is that in environments where there's a very dry winter, they actually avoid that winter by waiting to germinate until the spring. But in environments that have less variability in sort of wetter winters, they take this winter habit, which allows them to sort of continue to grow during the winter a bit. So these two different strategies that we're observing reflect differences in climate. So we're already seeing some interesting differences in relation to phenotypes, and now we're looking at the genetic basis of that. Similarly, days to anthesis, this is basically the same thing. We think of this as flowering time. What we see is that things that tend to come from hotter environments tend to flower much faster than things that come from cold environments. So, for example, if we want to think about applying this in our agricultural systems, if we can understand the genetic basis of this variation, it suggests that, if we want to adapt for hotter environments, it may be advantageous to find genes and variation of genes in these populations from that are adapted to these warm environments, which is what our lab is now currently working on. So interesting phenotypic diversity, and now we're starting to examine the genetic basis of that. So do stay tuned. We will continue to explore this. Okay, I promised we would talk about cassava as well. Cassava is a fascinating crop. It was domesticated in South America, and it's now grown throughout mostly the southern hemisphere. It's a staple crop in South America, Africa, Southeast Asia. But if we look at this figure of production over time, we see that obviously in places like Colombia, there's been, we had this, we won't talk about the green revolution, kind of changes in some genetic practices and some agronomy. We see this huge increase in yield per hectare, but now recently we've sort of plateaued. In some places like Nigeria, we're even seeing reductions of yield. Whether this is driven by climate change is not entirely clear, but it does raise some concerns that maintaining production of staple crops like cassava for a lot of these communities is going to be essential for food security. So our lab is interested in understanding how cassava adapts to climates. So what is the basis, genetic basis, for example, of climate adaptation in cassava? We have a project in collaboration with the International Center for Tropical Agriculture. This is a facility that is located in Colombia, in Palmyra, Colombia. They have a gene bank that means that they have, in the background, you can see these little test tubes. These are all different cassava plants that are maintained. They have to maintain them live in these collections. But these collections, each of these represents a plant that was collected from across a diverse environment. So imagine those images of someone collecting wheat. Well, someone also did this with cassava, but in this case, we're interested not in wild species. So this isn't a wild cassava, but we're studying what we call land races, which are basically traditional varieties that have not really undergone any intentional breeding, but simply reflect varieties that are grown by different people in different environments. I've drawn this little map. This is Colombia. This is the country of Colombia, and each of these points represents one of the locations at which a cassava plant was collected that we're studying. The color tells us about the temperature. This is actually the maximum temperature experienced during the year. If you look at this little plot here, you can see that we have varieties that come from, the difference in temperature is actually extraordinary. So this is obviously in Celsius, but we have a 20-degree difference in the maximum temperature. So we have things that come from much colder environments and much warmer. And this is because Colombia has a very strong elevational gradient. So there's places that have very high mountains, and then there's coastal regions that are basically at sea level. And so the variation in temperature is largely attributed to adaptation along elevational gradients. Similar to those experiments I was discussing about in California, from the coast to the Sierra Nevada mountains. So these varieties, cassava was domesticated thousands of years ago. So this really, with this variation across climate reflects the process by which these land races, these traditional varieties have undergone. We can think of it as analogous to natural selection, evolution. They've evolved. So the varieties, in fact, there's experiments have been done. If you try to grow one of these varieties from a mountain that typically grows in a mountain down in the coast, it really doesn't do well, and vice versa. So there's this adaptation, but we know virtually nothing about the genetic basis of this. So this project that we're working on with CIOT, we're taking these lines, we are sequencing the whole genome, and we've done this now. We have genomic sequencing for all these individuals. We're looking at genetic variation, and in this case, we're actually trying to find cases where genes are knocked out by mutation. By looking at climate, so here's that elevational gradient I was talking about, which you can see in Celsius. We use the climate to look for alleles, so genetic variation associated with climate. And then in collaboration with people at CIOT, we are then applying CRISPR. So you may have heard of this technology, CRISPR-Cas9, as a gene editing technology. It allows us to make targeted changes to the genome, where more or less wherever we want. Our collaborators at CIOT have really pioneered this and gotten this working in cassava. So now we can find genes from these populations, use CRISPR. So this is a picture of a little tissue culture. So to do this, we often grow these things called calli. So that's what you're seeing. This is a petri dish of cassava, but we can gene edit these and then grow these into whole plants. So you can see this is now a plant emerging. So if a gene edited one, we create. So we do things like we make these little genetic changes, and hopefully the idea is that we are identifying genetic variation that has contributed to the ability for these plants to adapt along this climate gradient. So this is the project that we're working on in cassava. It's an exciting project because it brings together a lot of diverse people and groups spanning UC Davis across different departments. So we're sort of thinking about genetics, climate modeling, and at CIOT, we have this diversity and also these transformation doing these gene editing approaches. So again, stay tuned. A lot of these projects, as a new faculty, a lot of these projects are new. So giving you a sneak peek. Okay. The last system I'll talk about that we work on in the lab is pistachio. Again, it's a very important crop for California agriculture. 99%, essentially, 99% of all the pistachio grown in the U.S. is from California, and California is the largest producer globally for pistachio. So pistachio and other nuts have an incredibly low carbon footprint. So if we think about how to feed the world sustainably, how do we produce enough protein, for example, to feed people? and we look at something like the amount of greenhouse gas emissions per 100 grams of protein produced. I'm sure this won't be news to people, but of course, a lot of animal products tend to have a high carbon footprint. And if we look at the very bottom of this list, nuts have the lowest carbon footprint. So in terms of producing protein, nuts are by far the most carbon efficient method to do this. This is for a number of reasons, one of which is agronomic. Nuts are grown on trees, which are long-lived perennial species, which means that we don't have to till the land every year. They are planted and then grown for multiple decades. So it's a very sustainable way to produce, sustainable in the terms of carbon output way to produce protein. Here's the challenge though. Pistachios and a lot of other crops, including many other nut crops, fruit crops, they require a cold winter. So here's a picture of an orchard. You know, we have this frost on there, and this type of experience. So plants, these pistachio trees, they need to experience cold winters. They have to go through a period of cold. It's called vernalization. It's required for them to properly flower in the spring. So this dormancy period during the winter is an important trait in crop species like pistachio. And we should be concerned about this because remember, people are planting pistachio trees now that they hope to be productive for the next 30 years. But if we look at climate models, it's clear that the amount of winter chill hours, so the amount of time that different, this is a map of California. We've kind of, this is the Central Valley. It's been rotated here. So if you look at this map, the color represents the number of hours that these regions experience of chill, which is something approximating the number of hours below 45 degrees Celsius. I'm sorry, Fahrenheit. But what we can see is that over the next decades, the number of chill hours is going to be dramatically reduced throughout California. And if you want to grow pistachio trees, this is something that people are concerned about because we want to make sure that we can produce pistachio even when there's warm winters. And so way we're approaching this, we're working in collaboration with breeders and researchers to try to identify pistachio varieties that can tolerate these warm winters, understand the physiology of this, the genetic basis of this, et cetera, so that we can prepare the pistachio growing industry for these changes in climate that we expect in the future. And so these are the predicted models that happened. This was published in 2009, and it turns out, and then indeed, it wasn't long before we started seeing some of the consequences of this. This is a picture from 2020, but also in 2015, there was really warm winters. The consequence of this has been inadequate leafing out. So here we see pictures of these pistachio trees that aren't leafing out entirely. Another consequence is that differences in the date of flowering between male and female trees can be asynchronous, meaning that there's no pollination, so there's no fruit production. So these changes in these warming winters are something that this is very, we should definitely be thinking about this for the maintaining the production of sustainable protein sources like pistachio. So that's the bad news, but I'm optimistic that there may be some solutions going forward. Here's a image of pistachio collections. These are just points from this something called the Global Biodiversity Information Foundation. This is pistachio trees. This is pistachio is native to like this region of the world. And what's important to note is that these different locations come from very diverse climates. So I've drawn a little map here, this is the distribution of observations of pistachio. And we've got the minimum temperature of the coldest month. So this is some estimate of how cold the winters tend to be. These orange lines tell us these are places in California currently. And as we've just seen, this is shifting this way. So what we're looking for is, well, we should be somewhat optimistic because there are varieties of pistachio that come from environments that are much warmer than others and warmer than what we currently have in California. So this is by studying the genetic variation in these varieties from warmer places, we might find stuff that's useful for preparing pistachio for the future. Here's some pictures of different pistachio varieties. I've shown these because these are ones that we're currently working on. We have both male and female trees if you're interested, but what we're really interested in is this variation in the timing of dormancy requirements. So how quickly these plants release from dormancy in the winter is a proxy for how much chill requirements they have. So by examining this type of diversity, we're hoping to gain insights into the genetic basis of changes in phenology. So for example, here's a variety Kerman. This is commonly grown in California. And here's another, so it has the way you can read this is these are the dates. And you can see this is the timing at which we see it releasing from dormancy. And then we have varieties like this one, Mature, which has an earlier phenology, which is, we're currently investigating this as a potential indication of this having lower dormancy requirements, meaning it needs less chilling hours in order to properly complete its winter vernalization. So what we're currently developing in this system is we've taken some of these lines, we're sequencing their whole genomes. We're using some more sophisticated approaches. I won't get into the details that give us really clear insights into things at the genomic level that we really haven't seen before. Here's a snapshot of this is a work in progress for sure, but what's exciting is that we're having the whole genome. So here's the genomes of pistachio. We have 15 chromosomes. And these are the different varieties. And so we can compare them and look for differences between them. So for example, by comparing the genome of Kerman versus this other variety, Mature, we might gain insights into what makes Mature capable of flowering much earlier, having lower dormancy requirements. And so we can expand this into all these different varieties as well. So we're excited about starting to look into these differences as a way of gaining insight into variation in phenology and winter chill requirements in the species. So these are a subset that we're developing these very high resolution genomic sequence maps. But we're also expanding this to looking at a large population. This is a really powerful resource maintained at the Wolfskill Experimental Orchard. Here's a picture, an aerial photo of this location. There's over 700 trees there, which are all largely genetically, there's lots of genetic variation maintained in this population, in this collection. This is similar to the USDA collection, et cetera. These were collected around the world. They were brought back here and then grown in this common garden in this environment in Wolfskill, which is just down the street in Winters. And so we're looking at this variation. We're now in the process of starting to catalog all of the genetic variation in this population and looking at trait variation to help us better understand the genetic basis of things like dormancy requirements even more and contribute to the ongoing breeding efforts to make pistachio more climate resilient. Okay, so I showed you projects we did in natural species. I showed you projects that we've done in wheat, cassava, and pistachio. Again, all kind of works in progress. But what I hope I've left you with is a sense that convinced that natural diversity in the age of climate change is really a natural resource. Being able to catalog the diversity that exists in species from different climates provides a glimpse into how evolution has solved the problem of climate adaptation. So, different species, different populations of the same species have all figured out a way, they've all adapted through evolution to these different climates. And so within all of that variation, there's valuable information that can be gleaned by growing these different organisms, by sequencing, looking at the genetic level, this variation. And so this diversity is something that we should really see as something that's valuable. And as a resource, we should seek to properly understand it, protect it, and use it if we want to address the challenges of climate change today. I'll leave you with one more thing. If you want to do something, if you want to contribute to any of this type of work, I would encourage you to go check out this app, iNaturalist. I took this screenshot last night. And here's a map. These are observations that people have made. You can get your cell phone, you can get the app, you can take a picture of a plant that you see, and This is incredibly powerful. It uses artificial neural networks, machine learning it will help you identify the species, and you can catalog the location. So throughout this talk, I've shown you lots of different examples where we're looking at variation of plants across different climates. And these observations have actually been used in science. People use the observations that citizen scientists make by going and making observations about species from different environments, cataloging it. It provides data into really powerful data sets to understand where species are found across environments, how they look, how they're different. So you can contribute if you want through things like this, Ad Naturalist. You can download it, go take pictures, learn something cool about the species in your environment, and also and contribute to the science that's going on. So with that, I'll stop now. I went a little over, but I appreciate the time and appreciate everyone's attention. Thank you.
Great, thanks. Thanks, great, that's wonderful. We have about 10 minutes or so for questions from the group. And if you do have a question, please raise your hand. I think there is a reaction function there. Any questions from the group? I know, Gray, you covered a lot, so my folks need a couple minutes to process. Maybe as folks are sort of thinking through this, I have a question maybe to get us started. I appreciate, Gray, you talking through all these wonderful tools and technologies that are out there. As somebody who's not quite familiar with this space, but works much more closely with community members and policymakers, do you have quick examples just off the top of your head of some of these technologies being taken on by decision makers? And have you seen them make any impact in sort of where they choose to grow, how they invest in different crops or technologies, or changes in rules and regulations or behaviors? Just a couple examples.
Yeah, I'm trying to think of like policy. I mean, I certainly, in the world of plant breeding, I mean, if you're a grower and you want to get a variety of a crop that's best suited for the climate, you might look to a private company that's breeding. We also have public breeding, like here at UC Davis, the pistachio breeding program. So we have Dan Parfit on the call. He and others have worked on this. So if you want to get a new variety, then you're using genetic variation that has been informed by a lot of these technologies. If you're growing crops, certainly important in that space to connect growers with the optimum genotype for the environment, and for future environments. Like if you're growing a long-lived perennial like pistachio, it starts to really matter because you're planting something. It's a 30-year or investment, in 30 years in the world of climate change is like not insignificant. So the genetics really matter there in breeding and variety improvement. I can't speak to the action that's happening in this space of biodiversity like conservation, but it's something that certainly crosses it gets brought up in these conversations of studying genetic diversity to identify endangered species, threatened populations. Like say you want to try to save a population that's declining of some endangered species. You might use some information about genetic diversity to make the decisions about where you're going to move that population or what new genotypes you want to introduce. There's lots of, understanding how adaptation works and knowing the genetics of it actually can be valuable for making better conservation decisions. I know that's something that people certainly is part of the conversation, definitely. Yeah, but I can't think of it. I mean, I guess in terms of policy, one policy point that's probably worth making is that, I talked about at the end in cassava, for example, like gene editing, like people are doing gene editing, starts to open the conversation about GMOs, et cetera. And where does that fit? Because that is largely a policy-based decision. So, what's our comfort level for saying if we can make sustainable agriculture, but it's going to require some GMOs? Are we comfortable with that? What's the regulatory landscape around that? very interesting conversation about trade-offs between sustainability and agriculture, which can be achieved by GMOs in many cases, but also balancing that with people's, thoughts about those things. So that I think that's an interesting area where policy and genetics has like a very tight, relationship.
Yeah.
I see Dan Parfit, you unmuted yourself. Go for it.
Yeah,
please.
I'll make a couple quick comments about that. Policy-wise, something that's pretty important actually is our germplasm collections and understanding and the extent of diversity and what kind of diversity are in these collections is pretty important. In pistachios, for example, we have very poor collections, and one of the problems is that we cannot go and get new materials very easily, particularly with respect to this Convention of Biological Diversity now, which has been enacted, means that a large number of the countries that we used to go to get materials involve a lot more bureaucratic negotiations, and I think as well as conflicts that are occurring in many of these countries. And so I think it's really important to understand the existing diversity that we have in our present collections. It's
Yeah, I think that's a really good point. Yeah, so both resources to invest in maintaining these collections, because I mean, if you lose a tree in your collection, I mean, it's gone. You don't have the genetics anymore, and that's something we should care about. And then the other point that I think Dan made that's really valuable to emphasize is like the rules and regulations around getting diversity from other countries, like what can be brought to the US and like what you can collect with permits. I mean, a lot of these collections I mentioned, like they were done in the 70s and there wasn't like, so now there's a lot more regulation about, going somewhere and collecting seed and bringing it. So, that's an interesting regulatory landscape that has emerged in the last decades that has changed the way in which we think about collecting and cataloging genetic diversity. But I think, I guess the point that's domestically relevant is funding for, maintaining these collections, cataloging the diversity, et cetera. I think is super valuable and worth emphasizing for sure. So thanks, Dan.
Wonderful. Thanks. Additional thoughts from the floor? Give it another minute or so.
Well, if everybody gets iNaturalist, go get iNaturalist, go collect, go make observations, contribute to the science. It's very cool. It's kind of fun. You can like learn about different species. And also, I mean, I know we've actually used this data from this in our own research for a different system. I didn't talk about it, but yeah, it's cool. It's very powerful. It's very useful.
Yeah. I might do that.
Yeah.
All right. Well, if there's no more questions, maybe I'll just give a plug for Gray. If any questions do emerge that you think of later on, I'm going to volunteer you, Gray, here for folks to email you and reach out. There's just so much that you're doing in your lab, and everything looks fantastic, and so hard to sort of comprehend and process everything all at once, but super interesting nonetheless. And I'm really glad that we got to hear about your work. And I really thank you for spending the time telling us about that, and for everyone to being here and listening. We were just in terms of closing remarks. I will say that Gray was very gracious in kicking off our winter seminar series. We have a lot more sessions coming up in case folks are interested. Next week, we have representatives, good friends of ours in the California Department of Public Health, the section head for climate change and health equity. And so we have Dan Wu and Abigail Ramirez coming in next week and getting that sorted out. And then after that, we have additional folks, including other faculty members from UC Davis, and we have deputy secretaries from different California departments coming through. So I'm pulling all the strings here to try to get folks to come. And so thank you, Gray, again, for kicking us off here this quarter.
Yeah. Thanks so much for organizing.
Yeah.
Looking forward to the rest of the quarter. I'm excited to see the rest of these seminars.
Great. And so look forward to those announcements. We'll be sending out email messages, announcements. Sophia will be doing that. So look out for those, and please stick with us and register. And thank you all again for being here. And Sophia, just about to share that information in front here. Yeah, future events, right? And then maybe Sophia can end with our social media contact information. Everything you need to know is there. And so thank you so much. And I hope everyone has a great week and a great day. And thank you again for joining us today. Thanks.
Thank you.
And thanks, Gray. Thanks.
Thanks, everyone.